Hello everybody, I thought I'd do one without my face on for a change. My makeup person isn't available, so we are without a picture of me. Anyway, hopefully it'll be less distracting to me. Anyway, we're going to look again at this genetic diversity within a species. Last lesson we looked at measuring that in terms of proteins in a gene. It also mentions in the syllabus that we need to do this as well, that by comparing the frequency of measurable or observed characteristics. When it says an observed characteristics, they're talking about a phenotype. Okay, and one that they like to ask about, especially when we do our year 13 work, is blood groups. Especially if it's human focused, because it shows it's really good diversity. There's 8 different blood groups, and there's 2 different systems. There's the ABO system, so you can be blood group A, B, AB, or O. And then there's the rhesus system, where you can be rhesus positive or negative, and therefore when you add it all together, there's 8 different blood types. I'm a positive person. Literally a positive person, only at certain times of the day though, in terms of my positivity. I'm obviously A positive blood all the time. And it's just interesting to see the distribution of different blood groups. The UK has the highest number of O negative people in the world. We have a large population, 65.5 million people. This is from Wikipedia data. So, yeah, how reliable it is down to how good Wikipedia is, but it tends to be good for stuff like this. And we've got 0.09 frequency. So I'm saying 65.5 million people equals 1, 0.09 of them, or 9% of them, are all negative. Okay, that's the same as Spain, and the same as New Zealand, but we're a bigger country than Spain. We've only got 46 million, and New Zealand's 4.5 million roughly. I've kind of highlighted like the top 3 or 4 each time, just to kind of show you, how it's spread around the world. And you could take a country like Ethiopia, where humans first evolved, that's where our early evidence for human remains are, and they've got 39% or plus. Go to the other side of Africa, to Nigeria, well, it's 51% or plus. And there's so much diversity, genetic diversity within Africa. In fact, there's more genetic diversity within Africa than there is in the rest of the world. So when people class people's ethnicity as Black African, it's just based on skin color. It's there's so much diversity within them that you could kind of see differences in. But we just arbitrarily judge people on colour, which is kind of topical at the moment. I mean, it just seems crazy that we just go on skin colour to kind of put us into groups. It seems bizarre to a biologist, because it depends what traits you are measuring, what phenotype you are measuring, and if you just go on one of them, skin colour, it seems very, very strange to kind of just go on one thing. You may as well go on like tongue rolling or blood group. So that's just to show the difference between them. And, it's interesting as well that in Japan there's no negative blood. There'll be the odd person, however, it doesn't show up to within my 2 decimal places that I've got here. And same in China. There's no O negative blood in China, because that was the pathway that humans took out of Africa. Some went east and some went west. Some stayed in Africa. Some, so humans spread out, and it must have been those who turned left out of Africa and went into Europe were the ones that some of them must have had a rhesus positive mutation, or maybe those who turned east out of Africa just didn't happen to have the O negative, the negative rhesus negative antigen. So it is really interesting, and I've just kind of put a few countries there. You can pause it and have a look, or go on Wikipedia. They also break it down to ethnic group on there, but they don't bring in the positive and negative. So you can see difference between Romani ethnicity, which is, which kind of affects Romania. There's lots in there, lots of people there, and hence why they're quite high for the is it B positive? I think I can't remember what it is. Oh no, anyway, there's one blood group that's common in certain groups. Just to kind of give you a bit of genetics, because we do more of this next year when we do about inheritance. There's two different genes for blood group. There's gene I, and they've arbitrarily called it gene I, and then there's the RH gene. So there's two different genes. We're not saying that these are alleles genes. Gene I is found on a different chromosome pair to gene RH. There's two different genes. The I gene has three possible alleles. Three alleles, not two, because we're used to dominant and recessive, aren't we? But here there's three. But I've only got two chromosomes, so I've only got two. So there's lots of, there's in a population there's three alleles, of which I will have two of them. So e.g. a person can be, so the gene is denoted by I, and then the alleles are kind of put superscripts. So this person would have AB blood. They've got a big A allele and a big B allele, and it's what's called codominant. Therefore, both are expressed. This person's got an A and an O. Well, O is recessive. Therefore, this person will have A-type blood. This person will have O-type blood because they don't have an A or a B. So you can be, and there's other ones as well. You can be IA, A-type blood, IB, B-type blood. You can end up singing the O MacDonald as well. It's, IAO. There's lots of different varieties. The other gene is the rhesus gene, and that is like GCSE, where you could be homozygous dominant, heterozygous, or homozygous recessive for it, and the big R, the dominant allele, makes you rhesus positive. Okay, so there's two different genes, hence this phenotype. It's complicated. I've got either an A, B, or an O system, and I've got a rhesus positive system. So these red blood cells have got basically two different classes of proteins on them. As I said before, I'm A positive. You can buy a kit on Amazon, or if you ever give blood, they'll tell you your blood type. So I might be IAIA, big R, because I've got an A and I'm rhesus positive. But I could be IAIO and big R, or I could be IAIA and then heterozygous for that one, or I could be A, big O, and heterozygous. So there's four possible genotypes that I could be, and I can look into my family tree and I can work out which I am, because if my mother's got O blood, then I must have got an O from my mother, and therefore I must be either this one or this one. And if my mother's O negative, then I must be this one. Again, things we do next year. Just to stress, we don't need to know about blood groups. I'm just kind of telling you about them because they can do the same in a question. They can tell you about stuff and then ask you about it. So, but we're straight into year 13 a bit in terms of the genetics, dominant and recessive and things. Person who is O negative has the genotype IO, little r. They have to be. If you had an A or a B, they'd be A or B blood, and they are homozygous recessive. So, last thing, how does a person with a big R allele make a rhesus protein? I thought we'd just zoom through this just to recap. Hopefully this is a nod along. As I say in class, I'm actually nodding and you can't see me, but never mind. So the rhesus allele is transcribed by RNA polymerase. Might get 2 marks for that, one for transcribed, one for RNA polymerase. mRNA is made and binds to ribosome. Might even get 2 marks for that. mRNA being the product of transcription, ribosome being what it binds to. tRNAs carry specific amino acids and bind to the mRNA. That's true. This is into translation now. Hydrogen bonds form between complementary anticodon on the tRNAs and codons on the mRNA. Yes, they do. Always mention anticodons and codons. The ribosome advances one codon at a time, forming peptide bonds between the amino acids, and then bonds form between R groups of specific amino acids to form the rhesus protein. I could slot in any protein here, and they normally slot in enzymes. But again, I thought that it's a good chance to just go through this. So, there's no real notes to write down for this. I've told you about it in an exam, or if we're in class, we'd measure some some of our characteristics, and we'd probably just do some kind of like scatter graph work on it. But quite frankly, again, we meet this again in year 13 when we get onto the genetics of it anyway. So yeah, it's just be aware that we can measure a phenotype of the same species, and I will see variation in it. If the organism has undergone stabilizing selection for that trait or characteristic, there'll be not a lot of variation there. Okay. If the population is undergoing selection for it, you'll get bigger variation in that characteristic. So it's just showing that. And the UK data for blood groups, it's very spread. It's O plus is the most positive, A plus is the most common, A plus is the next most common, but we are no real massively different to others. You can see that the US below us, and there's a slight difference. We have more negative blood, and you want in a population the most O negative people because they're a universal donor. Their blood cells have no A antigens, they have no B antigens, and they have no rhesus antigens. They're like naked red blood cells. So you can pop them into anybody and you won't have an immune response. The blood won't agglutinate. And therefore, you're the universal donor if you are O negative. You're the universal recipient if you're AB plus, because your body will not react to an A antigen, a B antigen, or a rhesus positive antigen because your blood cells are smothered in all three of them. But yeah, if you're AB positive, you can't give blood to other people. So it's normally quite, if you want to do some wider reading, blood groups, really interesting. Yeah, it's really interesting. And do cats have blood groups? Who knows? You could maybe find out. I'll stop there because I'm getting into like repeating Praspeldinion mode.